Video tutorial for microbiology with Sharnik. So in this video tutorial, or I should say from this video tutorial, we are going to see the polymerase chain reaction technique. Although I have already made a couple of videos regarding PCR, so today in simpler words we are going to revise the entire PCR technique. So what is PCR? PCR, the full form of PCR is polymerase chain reaction. So as the name suggests, polymerase chain reaction, that means with the help of polymerase, here we are performing a chain of reaction. Okay, so what kind of polymerase? That is the first question. So here the polymerase that is commonly used is Taq DNA polymerase, whose optimum temperature is near about 74 degrees centigrade. At that temperature they can show its enzymatic activity. Now the question is, what is PCR? Why it is important? So PCR means I have told you that polymerase chain reaction. So it is, in simpler words, in vitro DNA amplification method, or in vitro DNA amplification process, I should say. So PCR is DNA in vitro DNA amplification process. So that means here we are not using any kind of host for our DNA amplification that we are using in case of traditional DNA cloning. So that's well. So what kind, now let's come to the topic, what type of ingredients we need and to perform a perfect PCR. Okay, so first of all we need to have a DNA template. Remember, here there is a basic difference between the PCR and normal DNA cloning method. DC here for DNA cloning. What is the difference? Before the DNA cloning, before the DNA amplification with the help of traditional DNA cloning procedure, remember we have to treat our sample in which our gene of interest is present with restriction endonuclease, right? And with the help of this restriction endonuclease, we will simply cut out our gene of interest from the entire sample. Now with the help of vector and all that stuff, we will insert them into a suitable host. But in case of PCR, there is no use of restriction endonuclease. Okay, the entire DNA sample in which your gene of interest may be present or might be present is amplified with the help of PCR. Okay, so that is the basic difference between PCR and normal DNA cloning. In case of DNA cloning, you have to use a restriction endonuclease digestion to perform the further DNA amplification, but in case of PCR you won't require this, right? So the first thing that we require is the DNA template. Okay, that is the DNA segments to be amplified along with the along with the other DNA. That means our DNA sequence of interest containing source. Another thing that is important, that is the two DNA primer will be required. One is forward primer, another one is reverse primer, and it is a short DNA sequence which is exactly complementary to the 3' end of the template DNA. And remember one thing, that use of primer is very important in case of PCR, right? Because remember, here we are not using any kind of restriction endonuclease to separate only our gene of interest, right? So how can we only amplify our gene of interest without or less amplifying other unwanted DNA molecule with the help of PCR? So in that case we need DNA primer. Okay, not only that, remember, as it is a polymerase reaction, and we all know that in case of normal replication procedure, the primase enzyme provides primer and DNA polymerase simply extends it, right? So in that time, the nature naturally provides the primer, as it was a natural process, DNA replication. But here we are performing the entire replication, more or less we can say, in a thermocycler, that is the PCR machine. I am going to talk about the thermocycler within a minute. In vitro, right? So we have to provide the primers also artificially, so that the Taq polymerase that plays here the role of DNA polymerase can extend the 3' wedge group of the primer. That is the reason we need two primers. Okay, one is forward primer and another one is reverse primer. And most importantly, a Taq polymerase, a enzyme is used. Okay, then a suitable buffer solution, important for making the environment suitable for the chemical environment, any kind of enzyme substrate reaction, because polymerase is enzyme. Okay, we need divalent cation such as calcium, monovalent cation such as potassium ion for enzymatic activity, as we all know that we need such kind of cation for performing enzymatic activity. And the most importantly, the thermocycler machine, or in general speaking, PCR machine. Okay, so what is the utility of this PCR machine? Let me show you the PCR machine first. So this is a thermocycler machine, that is commonly known as PCR machine. So what is the utility of that thermocycler machine? This thermocycler machine have the ability to change the temperature. Okay, because remember, in PCR, in each cycle there are 3 steps. One is the melting step, which is near about of 93 to 95 degrees centigrade. Another one is annealing step, which is of 50 to 70 degrees centigrade, depending on the TM of the expected duplex. We are going to discuss about this. And the last one is the DNA polymerization step, where we have to keep the temperature 74 degrees centigrade. So the thermocycler machine have the ability to change the temperature from 93 to 50 to 70 degrees centigrade, and then from 50 to 70 degrees centigrade to 74 degrees centigrade within some minutes, right? So that is why thermocycler or PCR machine is used for performing the PCR technique, although it was discovered far after the actual PCR procedure has developed. Okay, now let's discuss another important thing, then we will go to see the basic steps of PCR technique, that is the main topics of today, that is advantage of PCR over DNA cloning. Why PCR is advantageous than DNA cloning? So, and I am also going to discuss about disadvantages of the PCR too. Okay, so advantages of PCR over DNA cloning. So what is the advantage of PCR over DNA cloning? So remember that a traditional DNA cloning procedure with all the troubles and contamination take 2 to 3 weeks to complete, right? On the other hand, PCR can give you billions of amplified DNA of interest within 1 to 1.5 hours. That is the thing. PCR just saves your time in terms of DNA cloning. That is the reason PCR is more preferable if you are just willing to amplify a DNA molecule. If you want to see the expression, then PCR cannot help you. You have to go for expression vector to express your gene of interest, right? And what is the disadvantage of PCR? So there are some disadvantages of PCR too. As I have told you that PCR cannot express a gene, this is the major disadvantage. And another important disadvantage in case of PCR is we cannot use the PCR technique for unknown DNA sample, okay, whose sequence is not known to us. Because remember, for performing the PCR technique we need primers, right? And primers is nothing but the complementary short segment of DNA which is exactly complementary to the 3' end of the DNA template. And if you do not know what is present in the 3' end of the DNA, then how can you make the primer, right? So that is why PCR process of unknown DNA sample cannot be possible. On the other hand, you can clone DNA, a unknown gene of interest. You can amplify a unknown gene of interest with the help of DNA cloning. How? You will simply suppose this is the unknown DNA sequence. We all know that we have an enzyme called terminal deoxynucleotide transferase. If you add terminal deoxynucleotide transferase in presence of DATP, then terminal deoxynucleotide transferase simply add poly A nucleotide in the 3' end template independently, right? Now on the basis of this AAAA, you can generate TTTT, or otherwise there is more easy procedure that is present, that this is a unknown sequence, that is you are willing to clone. What you will do, you will simply add DAM methylase and DCM methylase, because we all know DAM methylase and DCM methylase are methylating enzyme, right? So it will simply methylate your gene of interest. Okay, then you will simply add your linker. What is linker? Linker is a short segment of DNA molecule which is nothing but artificial recognition site for enzyme. Suppose you have used recognition linker of EcoR1 and you have also methylated it, so EcoR1 cannot cut your gene of interest if there is any recognition site present, but it will simply cut your linker. Okay, and by that method you can easily insert it into a vector and transform it into a suitable host and amplify them. So that is the idea how unknown DNA sequence can be cloned. But in case of PCR, you cannot do nothing without the generation of primer. That is why for unknown DNA molecule amplification with the help of PCR is not possible. And another important thing for short question, that after the third cycle of PCR, you can have a purified gene of interest in its amplified version. What does it mean? It simply means, remember, you are using the PCR technique when your double-stranded DNA molecule is something like that, right? That means it is the unwanted yellow color one is the unwanted DNA source, and among them your gene of interest is also present. Now you will simply melt them, which is the first step of PCR technique, and you will generate a primer specifically of the 3' end, okay, in both the side. So DNA polymerase will extend just like that. So as you can see, along with the amplified gene of interest, there is also some unwanted material is also present. So after the third completion of third cycle, you will have a purified amplified version of gene of interest. That means you will have this kind of things after the end of the third cycle. This is very important question for MCQ. They can ask you that after what stage you can find purified gene of interest in amplified version. Okay, so you have to just tick on the end of the third cycle. Okay, so now let's enter the main steps of the PCR. So as PCR's name suggests, it is a chain reaction. What does it mean? It simply means that the newly synthesized DNA molecule that is generated by PCR will again, or I should say that is generated by one cycle of PCR, will again take part in the PCR as a template for further synthesis of DNA molecule in subsequent cycles, and in that way the cycle goes on up to infinite time, unless or until you switch off your PCR machine, right? So remember, in each cycle PCR has 3 steps. Okay, one is denaturation step, another one is primer annealing, and last one is DNA synthesis or primer extension. So let's focus on denaturation. So in denaturation, wait a minute, that you can simply take a screenshot of this. I am going to explain you the entire step with the help of a diagram. I think it will be very useful to you rather than know the entire thing theoretically. So let's start. Okay, now so we have to add building blocks. That is also important requirement for PCR. Another one is primer. We have to add polymerase. Remember one thing is it is tagged DNA polymerase along with the DNA with the gene of interest. Remember here the sky colored portion is your gene of interest and the gray colored portion is the unwanted part. So the first step is the denaturation step or melting step, which is for mammalian genome is near about 93 to 95 degrees centigrade. Okay, so after denaturation the primer will bind. If I consider this portion is 5 prime, this portion is 3 prime, so here, and in that one here. Okay, so primer binding step is known as annealing step. And remember one thing, the annealing step is very important and a very crucial step of PCR technique, and it required temperature 50 to 70 degrees centigrade, which is obviously quite lower from the denaturation step. But we have to keep one thing in our mind: the temperature should not be too much lower, because there can be possibility of mismatch hybridization, which can only be prevented by using a temperature which is not too high for preventing successful hybridization of our primer with the 3' end of our gene of interest, but it is also high to prevent any kind of mismatch hybridization. Okay, now how can you determine the annealing temperature? So for that reason I am again going to come back in my notes. Let's understand one thing: the primer will bind in such a way. So the temperature of annealing will be determined by the Tm value of the primer. Okay, we all know what is Tm value. Okay, so the primer's actual temperature will be 5 degrees centigrade. The annealing temperature will be 5 degrees centigrade below the Tm value of the primer. In general, it is normally kept in between 50 to 70 degrees centigrade, again depending on the Tm value of the expected DNA duplex. That means the duplex formed by the primer and the complementary end of the 3' end of our gene of interest. Okay, so there are various equations that you can use to determine the annealing temperature. I am going to discuss with you a important rule, that is Waller's rule, that can tells us the exact Tm value of a perfect duplex of 15 to 20 nucleotide long. And remember one thing, that our primer is near about that of length, so we can use Waller's rule at high ionic strength 1 molar NaCl. So we can prefer Waller's rule for our process. So the rule is that one: Tm in degree centigrade is equal to 4 into G plus C plus 2 into A plus T. So A plus T here is the sum of A and T containing nucleotide, and G plus C here is the sum of G and C containing nucleotide of the primer. Remember one thing, not duplex. Okay, this is the A plus T and G plus C is only the number of nucleotide in the oligonucleotide primer. You have to keep this in your mind. Okay, so primer annealing as you can see, that primer annealing is very important step. We have to keep one thing in our mind, that the primer annealing temperature should be lower as compared to the denaturation temperature, so that our primer can hybridize with the 3' end of the complementary strand, but it should not be more lower, so that, or I should say it should be higher to prevent any kind of mismatch hybridization. Okay, so annealing temperature is usually kept 5 degrees centigrade below the Tm value of the primer. So we have also discussed a rule that by which we can conclude the Tm value of expected duplex, and with the help of this Tm value of expected duplex we can conclude the Tm value of the primer. So that is the idea. So after primer annealing, let's move to the diagram again. We are going to perform the extension step. Remember one thing, that this kind of extension is nothing but the extension of the 3' OH group provided by the primer by DNA polymerase, here tag DNA polymerase. This step is exactly the same step that happens in normal replication. In that time, DNA polymerase 3 plays the role of polymerase, and with the help of its 5' to 3' DNA polymerase activity incorporate DNTPs, that is the building blocks of the DNA, okay, depending on what is present in the complementary template. So that is why we have incorporated the DNTP. So that is how the extension process going on, and here the extension, the role of polymerase is played by tag DNA polymerase, and as its optimum temperature is 74 degrees centigrade, so after annealing we have to again increase our temperature to match the optimum temperature of that polymerase that we are using, right? So then it will simply extend the 3' OH group provided by the primer and synthesize a newly formed double stranded DNA molecule. Okay, now as you can see that it is a 3-step PCR, right? One, that means it's a PCR which contains 3 steps in its one cycle. So one denaturation step, one annealing step, and one extension step. Now there is a thing that you need to know. If the annealing temperature and the extension temperature is within 3 degrees centigrade, that means the temperature difference between annealing and extension is just of 3 degrees centigrade or less than 3 degrees centigrade. As you can see that the annealing temperature that we are using is 50 to 70 degrees centigrade and extension temperature is 74 degrees centigrade. Now just think, if the Tm value of the primer that we have obtained is near about 77 degrees centigrade. Okay, so as I have told you that we have to set our annealing temperature 5 degrees centigrade below the Tm value, so that now we can feel safe that there can be no chance of any kind of mismatch hybridization. So we have set our annealing temperature 72 and extension temperature is 74. So what is the difference between 72 to 74? Just 2 degrees centigrade, right? So under such circumstance, can we merge the annealing step and extension step into one step? Isn't it? So that is the idea that commonly used whenever the difference between annealing and extension temperature is just within 3 degrees centigrade. So in that case, our entire PCR become 2 steps, right? One is our denaturation step and another one is the mixture of annealing and extension step. So this kind of PCR is known as 2-step PCR. This is very important for university question. They can ask you what is 2-step PCR, then you have to explain this thing to get 2 marks. Now the newly synthesized DNA strands will again involved in PCR, and here they are playing the role of template and leads to the formation of 4 double-stranded DNA molecule. And after the end of 3rd cycle, as you can see, we will have our purified gene of interest in amplified form, right? But before that stage, you can see there is no gene of interest is in purified form. There is unwanted part, unwanted part, unwanted part, unwanted part, unwanted part. That means no purified product present. So that is the reason, if you want purified version of your gene of interest, so you have to wait for third cycle. In that way the PCR cycle goes on. Usually we run the PCR cycle up to 30th cycle, and after the end of 30th cycle we will have billion of amplified DNA molecule. Okay, and how much time it takes to complete the 30th cycle of the PCR? You will be surprised after knowing. Just 1 to 1.5 hour. Okay, that is that much fastest technique PCR is. Okay, so that is all about PCR technique. So let's see the graph again. So this is the denaturation temperature, which is denaturation step or melting step, which is for 1 minute. Annealing temperature again 1 minute, and extension temperature, okay, for 2 minutes. So this PCR technique is possible just because of tagged DNA polymerase, because tagged DNA polymerase is the only DNA polymerase, and there are also some kind of DNA polymerase that I am wanting to just say that those kind of DNA polymerase that whose optimum temperature is about 74 degrees centigrade, right? That means they are thermostable. That kind of DNA polymerase. Otherwise, if tagged DNA polymerase is not discovered, there is no existence of PCR. You have to remember this. Now the question is, from where tagged DNA polymerase is isolated? So remember that tagged DNA polymerase is isolated from a bacterium called Thermus aquaticus, which usually lives in geothermal vents, and as usual it is also a thermophiles, right? It lives in hot spring, and it possesses the tagged DNA polymerase which involved in replication of the Thermus aquaticus. So that's it for today, guys. This video is just the basic discussion of PCR. We will again come back with another video where we are going to discuss about the real-time PCR, quantitative PCR. Then we will discuss about the reverse transcriptase PCR, and in that way we will wrap up our discussion regarding PCR.